let's get started with part 2 of chapter 3. At the end of part 1, we were discussing carbohydrates and switching over to lipids and discussing how lipids are hydrophobic. That means water-fearing. So they don't dissolve in water, they are repelled by water. So for example here, like we've got what are called phospholipids, and we'll look at how those are important for our cells. The inside part is hydrophobic. That's the fatty acid tails. The yellow part is the phosphate heads that are polar, they're charged. They're hydrophilic, they like water. Water's the little red molecule here surrounding that. So what happens was when you put fats in water is they stick together. They make like fat droplets like this, where you've got all the hydrophobic parts together, separating them from water. Okay. So fat, we'll look at the triglyceride. So you've got a glycerol and triglyceride, 3 fatty acids. So you'll have these fatty acid tails that attach on. When we take in food, if we don't use those calories, they get converted into triglycerides. And one measure of health that they'll look at is your triglyceride levels in your blood. And it has to be under 150 milligrams per deciliter to be considered normal. Okay, so why do we need fats? We do. We need fats. First, energy storage. So when we don't use those calories, we store them. You can break the bonds, put them into cellular respiration that we'll look at later, and make energy. Also cushioning. Think about where you have the most fat. That's going to be in areas around our organs. They help protect and cushion those areas. They also help insulate. So someone who's really, really skinny has a problem maintaining normal temperatures. They can go from hot to cold really fast. We're also going to look at different types of fatty acids, saturated versus unsaturated fats. You've probably heard of these, but you don't know chemically why they look like they do. So saturated fatty acids means all these carbons here are saturated with hydrogens. There's no double bonds, there's lots of hydrogens. Whereas unsaturated have a double bond. So see, these two carbons have less hydrogens on them. And that also causes a kink or a bend in the fatty acid chain. So they don't sit on top of each other very easily. So saturated fatty acids, those really flat ones, the straight carbon chains, can stack on top of each other really nicely. They're generally going to be solid at room temperature because they fit together so well. Whereas unsaturated fats, those have lots of double bonds, bends, kinks in their chains. They don't sit on top of each other very nicely, and so then they are generally oils at room temperature. So most animal fats are going to be saturated fatty acids. Plant oils, low in saturated, they're more unsaturated. Okay, and another type of fat is steroids, and their structures are very different. They're not long chains. They are instead ring structures. So steroids are all derivatives of cholesterol. Cholesterol is the base, and then we change the structure slightly to make different steroids. Steroid hormones, for example, sex hormones, testosterone, estrogen, progesterone, your corticosteroids, your stress hormones, they're all important for your body, helping, to prepare it, send signals between different cells. Okay, so we've covered carbohydrates and lipids. Now let's move on to proteins. Proteins are the most variable in structure and function. They are going to be made out of what are called amino acids. So a protein is a polymer, a long molecule made out of the building blocks of amino acids. And different amino acids have different functional groups on there. They're going to look a little bit different. They're going to cause the protein to have its shape and function. There are 4 types of proteins generally, like structural, like keratin in your hair, storage for energy, contractile proteins, like in your muscles, and transport proteins. So like hemoglobin in your blood is a protein that helps us transport oxygen. So there are 20 different amino acids. And all of our proteins are made up of changing the order of these different amino acids. So you've got some that are hydrophobic, some that are like acidic, basic, hydrophilic, and how they're put together is going to determine how the protein folds. So what does each have? They each have a carbon atom with an amino group, this NH2 with nitrogen. And then they have a carboxylic acid, and then a side group. This side group is what changes amino acid to amino acid. And what they look like determines if they're hydrophobic or hydrophilic. So just like building the other polymers, you take the monomers, the single parts, and you put them together with dehydration, removing H2O to make this bond. And in proteins, this bond has a special name. It's a peptide bond. Peptide. So proteins made of amino acids bound together with peptides. So a small protein is actually called a polypeptide because it's got some peptide bonds, several. Your body has a very wide-ranging amount of different types of proteins, and the arrangement of amino acids is what makes them different. So you have different levels of structure. That first structure, like we had here, is the primary protein sequence. That's just the order of amino acids. So you can see this protein going down, and it's made up of all these different amino acids put together. And then you'll have a very specific order to determine the protein function. If you have even one amino acid that's wrong, like a mutation in your genetics that codes for the proteins, you can have diseases. So a great example that you'll see oftentimes is called sickle cell anemia or sickle cell disease. And there they just have a single mutation point on their sixth amino acid out of 146, and that changes what hemoglobin looks like. And instead of being a full structure, the cells will have this like sickle or like moon-shaped portion. They're smaller, they're not able to carry oxygen as well. It actually has really cool indications for like malaria countries, and you'll see this more often because it can help with that. We'll look at that more in later chapters though. Okay, so once you've got the primary structure, you'll have the secondary, tertiary, and quaternary. We'll look at those with hemoglobin. Okay, so primary. Secondary, you can have two different types. You can either have these alpha helixes or beta pleated sheets. And throughout the protein, it'll do different parts based on the amino acids there. And then the tertiary structure, you've got this polypeptide, this single part. And the quaternary is where you've got all the subunits, all these put together to make one big protein. Let's look at a nice example of that. So hemoglobin, we were talking about that. Let's look at a normal, healthy, functioning hemoglobin protein. So primary structure, the list of amino acids. Secondary parts are going to be alpha helix, the others are going to be the beta pleated sheet. And then together you've got this tertiary structure. That's one subunit of the hemoglobin molecule. So on there you can see you have this heme, that's where you've got the iron that binds oxygen. And then for a whole hemoglobin protein, you put together 4 subunits, 2 alpha and 2 beta, and each of those has a heme group to make one hemoglobin molecule. So that one hemoglobin molecule can bind 1, 2, 3, 4 oxygens. Primary, secondary, tertiary, quaternary. And how do we determine the protein structure? That's based on hydrophilic, hydrophobic portions. It's based on hydrogen bonding. You'll have these weak hydrogen bonds. You can make like these sulfur bridges between different parts. And then, if that protein is put in unfavorable temperature pH changes, so if it gets really hot or really cold, pH drops or gets too high, the protein unravels and denatures. It loses its shape. So there's the normal functioning protein. You throw it in a bunch of acid, that protein denatures. It unfolds. So like when you cook an egg, that egg white, turns white. And then when you cool that egg back down and you eat it, it's still white. It doesn't go back to before. Once it's denatured, that protein is done. You need to make a new one. Although they are doing really interesting research right now where they've found a way in at least one protein to take the denatured protein and refold it, which is really exciting. Okay, the last type of organic molecule we're going to look at are nucleic acids. So this is how we store information, send it on to new cells. You've got two types. You've got RNA and DNA. DNA, it's this double-stranded structure where you've got each side bound together, whereas RNA only is single-stranded. DNA is in the nucleus. That's the code that we pass on cell to cell. While the cell is functioning and it wants to make new proteins from this code, it turns it into RNA, and that RNA gets read and forms the order of amino acids for a protein. So this codes for the amino acid order. So we went through some of the differences. Another difference, DNA deoxyribonucleic acid, each of those has a sugar. DNA is one oxygen less on that sugar, so deoxy. Whereas RNA is made from ribose, which has an extra oxygen. So the parts, you've got a phosphate backbone, you've got a sugar. So in this case it's deoxyribose, so this is DNA. In RNA it would be ribose. And then you've got a nitrogenous base. That's going to be the coding part. So you've got A, C, G, and T for DNA. A binds with T, C binds with G, and you get that double-stranded helix. A, C, G, T. So you've got these purines and pyrimidines at these double ring and single ringed nitrogenous bases. And then they attach to the backbone of phosphates, and you've got this code. So you know on the matching strand that goes up with it, you're going to have an A, a T, a G, a C, and A. So that A binds with T, C binds with G. There you go. Now you can see those sides being put in. And then RNA is different in that the sugar is ribose now. And there's one different base. So here we replace the thymine with a uracil. There's no T in RNA, there's a U. So anytime DNA has an A and we read it, we put in a U instead of a T. And how we discovered DNA and its structure was really neat. There were a couple competing groups of people, scientists looking for the answer. And they didn't know if it had 3 strands or 2 strands and how they came together and how it coded for our protein. But they knew that somehow we were able to pass on information from parents to offspring. Because offspring, they look something like their parents, but we didn't know how until then. And we can actually use these DNA and proteins as evolutionary measures, looking at how closely organisms are related. There are some genes on our DNA that are the exact same in humans as they are in fruit flies, as they are in plants. And we can use those to look at how many changes have been made there to determine how close different organisms are related together. So things that are really important will have very little changes. And things that were more closely related, that had more common ancestors, will have more genetics in common. And they've used these to look at how different species are related. And so then you can make one of those phylogenetic trees and look at where everything branched off. This marks the end of Chapter 3.